Just hold on. Now it will be audible. Okay, now I think it's audible. All right, I think it's audible. You should have informed this earlier. All right, thank you so much for informing. I think now it's audible. All right, thank you so much for informing. Okay, so now we can start our session. All right, can you start our session right now? Okay, so the first question on the screen, this question is very fundamental question, and this data is given in your NCERT, and which is very useful and very important if NEET is concerned. Which of the following element is present maximally in earth crust as well as in human body? So this is the question. You can clearly see, and you have to say the answer. Okay, let me find how many of you are answering correctly. Okay, people, you have to answer this question. Student, you have to answer this question. Okay, I can see Deepti, Tina, Ornob, Ishsardi Creation. I think this is Rupsha, Tanmoy, Ritik, Ornob, Shorumita, Ahona. Yes. So I think now I'm audible to everyone, I guess. Okay, now please say the answer. What is the answer of this question? Which of the following element is present maximally? Okay, just let me tell you this explanation. Okay, because this is very fundamental question, so I think you should be very careful while you are answering in this question. What is the catch? The catch of this MCQ is that this is something element. Okay, this is something element that you have to say, number one. Another catch is present maximally in not only the earth crust but also in the human body. All right, beta. So now you answer. Okay, people are answering it, but there is a I am receiving mixture of answers. I can see people are answering nitrogen, few are answering oxygen, and I am damn sure that why people are making mistake here. So I think without any delay I should explain this question. So okay, all right, beta, don't worry, I will explain this. Okay, so if you have the idea, whenever you were saying this, we are not saying about the atmosphere. Okay, we are not saying about the atmosphere. If you say about the atmosphere, straightforward, we can answer this is a nitrogen, because it consists of 97% of the gases in the air. But we are not right now considering the atmosphere. The question is earth crust, the question is human body. Whenever you are considering the earth crust, if you have the idea of the metallurgy, your chemistry teacher definitely has taught you. So there is many compounds which are made of oxygen, na beta, like barium, silicate, aluminate, this type of compounds, already. Okay, so here if you consider element, then oxygen is maximum in earth crust. And if you consider the human body also, most of our human body consists of water. So there also oxygen is highest as per the element is concerned, and this data is given in your NCERT. Okay, so that's why it's very important for you. I will also show you at the end that how this question can be important, and how and where this data in NCERT is given, because I will also show you NCERT surgery at the end. Okay, before, let us solve the MCQ, okay, to charge up our brain. All right, the next question on the screen, the answer is oxygen. The next question is another conceptual question, and which is again given in your NCERT. The question is, all of the following amino acid can absorb wavelengths of light except. That's the question. Again, I'm repeating. I will definitely also explain the central concept of this question, but before, you have to understand that what the question is trying to tell. There are 4 options which are given. Three of them can absorb sunlight or any wavelength of light, but one cannot. You have to pick up that option which cannot absorb sunlight or any wavelength of the light. Okay, so people are answering. Okay, someone has answered A. Okay, let me find other answers. Okay, first answer first, beta. Answer first. If you know this answer first, otherwise I will start explaining. Okay, so no one is answering. You should answer, Ankita, Anindita Chatterjee, Ankita De, show me. Okay, all right. So I'm explaining the central concept of this question. Listen, the concept is this aromaticity. You know, I know you know the chemistry. In the organic chemistry, there is a concept is called aromaticity. You know, aromatic compounds. Okay. Now, Prime TV has answered A, beta. Okay, let me explain. Let us find whether Prime TV is correct or not. Okay, so PrepMed powers are answering A, some PrepMed powers are answering D. All right, nice. So this is aromatic, you know. So in chemistry, I think you have read the aromatic compounds are they have a particular property. That property, they will have proper smell, and at the same time they will have proper color. And the reason behind the color, that they can absorb wavelength. Okay, wavelength of light. Okay, so you know, like in these options, tyrosine, it's an aromatic. I think at least you have the idea that tyrosine is nothing but when you hydroxylate phenylalanine, you will get tyrosine. Tryptophan, it's again aromatic. Phenylalanine, obviously phenyl, its name containing phenylalanine. So the correct answer would be glutamic acid, because it is not an aromatic compound. So it doesn't have any particular smell, or it doesn't have any, you know, like color. And it cannot absorb the wavelengths. Okay, so the correct answer is glutamic acid. So correct answer is made by Ambika Das, SRD creation, Shayondeep. Okay, proud of you. Shoromita Malakar, correct answer. Arnav Hajra, correct answer. Ankita Ghoshal, correct answer. Shubhadeep Ghosh, correct answer. Ankita De, correct answer. Okay, so all right, so we can move into the next question. The next question is question number 3. Question number 3, listen, in current days, if we see the data are being changed as per the total number of amino acid is concerned, but we have to answer as per NCERT. Okay, that's why this question will be judged as per the information of NCERT. All right, so you know, the question is, which of the following is wrong with respect to the amino acid? Okay, so there are 4 statements which are given with respect to the amino acid. 3 are correct, but 1 is wrong. You have to find out the wrong statement regarding the amino acid. The first statement, 21 amino acids are there in the nature. Okay, though we have also discovered another amino acid which is called selenocysteine, though this is not included in 21, but still this is the correct statement for our syllabus, because NCERT has mentioned it. All right, so structure of amino acid changes according to the pH of the medium. Okay, this is a very conceptual statement. If you don't know the concept behind it, you will make mistake on the statement B. Okay, so I am keeping this statement as a hold, because I think I should explain a little bit for your revision, or if you don't know, I try to explain. Okay, so glycine is an alpha amino acid. Absolutely correct answer. I think you also know the structure of the glycine. If you know the structure of the glycine, it's you know, this is COOH, this C containing hydrogen, this C containing COH, and this same C containing NH2. So obviously it's alpha amino acid, because it's alpha carbon containing the functional groups. Okay, so it's absolutely correct answer. Glycine is your alpha amino acid. Next statement is valine is acidic amino acid. No, it's wrong. Valine is not acidic amino acid. Okay, I just want to tell you better. There is a you know, why I have chosen this question in the last year, though this question was asked from your organic chemistry, but still I want to tell you to remember a few things. You have to remember the list of acidic amino acid and basic amino acid. Okay, so before telling this, I want to tell you, you know, you basic amino acid, please do remember the list which is very popular, that is HUL. What is HUL? H for histidine, A for arginine, and L for lysine. Okay, and exactly last year, in the last year NEET exam, this question came, though I know it came in the paper of your organic chemistry, but it's important. Okay, so H for histidine, A for arginine, and L for lysine. These 3 amino acids are basic amino acids, and you have to remember this. Acidic amino acids are actually many, any glutamic acids and many acids are there which are acidic, but basic amino acids are few. That's why you can remember in this way. HUL is the examples of basic amino acids. So valine is And valine, I want to say you, valine is here mentioned as an acidic amino acid. This is again wrong statement. So so this is wrong. Okay, this statement is wrong. So correct answer is option D. Okay, because valine is not acidic amino acid. Valine is also a basic amino acid. Okay, it will come also in the statement of basic. Okay, valine. Okay, now I want to tell you the explanations of the option B. Okay, in both biology and chemistry, there were this question. Yeah, you are correct actually. So the next question is that how to explain the option B. The amino acid changes structure as per the pH of the medium. Let us take an example of structure of amino acid. This is C. This is, you know, like this is let us take hydrogen. Let us take, this is like, you know, this is NH2, and let us take, this is the R, and this is a COOH. This is the structure of an amino acid. So, you know, if the pH, what is the pH? pH is the hydrogen ion concentrations, that is the negative logarithm of hydrogen ion concentrations in the surface. So suppose this is the hydrogen ion concentrations. Okay, you know, this is the hydrogen ion concentrations. Okay, you know, this hydrogen would react with any lone pairs if they are. So what would happen, that in this NH2 will become NH3, and thereby the next thing which will happen, the structure will completely change. If your H plus structure, if your proton concentrations around in the medium is high, that would react with the lone pair present on the NH2 group. But if this hydrogen concentration is not that much, okay, so then you will find a different story. All right everyone, yes or no beta, let me know. Okay, all right, have you understood? Everyone please respond. If you are enjoying these sessions and if you are getting the concept, then please let me know. Are you getting this beta? Okay, are you getting this, yes or no? All right, so yeah, the pH, surrounding pH will decide the structure of amino acid, definitely. Okay, correct answer. Question number 4. Okay, absolutely correct. Question number 4. Okay, next question. The next question is which okay, Ahana is saying yes sir, Srikant Odikar is saying yes sir, Shohum is saying yes sir, and Rupsha is saying yes sir, Tina is saying yes sir. All right, thank you so much for responding. Okay, the next question on the screen, this is a very important question, and this question is based on the unique concept of the DNA. So if you have the proper concept of the DNA structure, then only you can answer this question, otherwise it will be difficult for you to answer. So let us find how many of you can answer this question. So the question is which of the following is mismatched? Okay, you have to tell the mismatched wala thing, that when it is mismatched. So 3 are matched, 1 is mismatched. You have to mark out the mismatched option. Okay, I'm giving you 1 minute and you ask me the answer, you tell me the answer. All right, okay, let me find how many of you are answering. Okay, please keep answering. All right, okay, keep answering. Okay, everyone keep answering. All right, okay, which one is a mismatch? Which one is mismatch beta? Please answer this question. Which one is mismatch? Which one is mismatch? Please let me know. Okay, let me find how many of you are answering correctly. Okay, all right. Okay, all right. Okay, so they are answering as D. Okay, so let me explain. First option, 5 3 and 5 prime and 3 prime. If I see 5 prime and 3 prime, okay, 5 prime and 3 prime end, it helps in the phosphodiester bond concept. Absolutely correct. Actually, you know that concept as well, where basically we are considering here our deoxyribose sugar, so for DNA. or if I say RNA or ribose sugar. So this is the, like, for it is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, and this is the fifth carbon, CH2OH. And you also know it contains these carbons 1 2 3 4, and this is the fifth one. It contains a functional group which is actually OH. Okay, it, OH. In the second carbon we lack one oxygen, rather it contains only hydrogen. That's why it is known as deoxyribose. If it contains OH, it is called ribose sugar, which is of RNA. Then again OH, the fourth carbon, it is a valency satisfied only, so only it will have hydrogen. And the fifth carbon, again if I say this is a OH. And if I say as per the reaction is concerned, this third wala OH and the fifth wala OH, this is very important to lengthen your chain in the DNA. Okay, so obviously all the reactions which will be in between two DNA, and it will be in between two nucleotides, this will be mediated by the 5' OH and the 3' OH. Though we know that in the 5' OH we have something phosphate group which is to be added, and then finally it would bond. But I just want to define that is, yes, the option A is absolutely correct. Option A is absolutely correct, that it is 5' end, 3' end, which helps in the phosphodiester bond, which is nothing but the backbone of DNA. DNA and RNA, for both backbone is 5', 3' phosphodiester bond. Okay, next question. In C terminal is a peptide bond. You also know the same concept if you have seen the amino acid structure. This is the carbon, this is the NH2, this is the COOH. This is COOH, this is like R, this is like hydrogen. This NH2 will react with the COOH of the next amino acid, and thereby ultimately you will get a bond which is called amide bond. This is called NHCOO bond, and this NHCO bond is called amide bond. All right, so and finally at the beginning one amino acid will be free, at the end one C terminal will be free, and at the beginning one N terminal will be free. So ultimately you will get one N terminal, you will get one C terminal. Okay, so this is a concept which is valid for the protein and the peptide. Next is the base pair. If I say about the base pair, the base pair will form is actually nothing but hydrogen bond. If I say for the DNA what is actually base pair, if I want to explain, I think most of you know that this is nothing but base pair. So let me explain again. So this is if I say this is the DNA, in the DNA there is base pair, and these base pairs are nothing but hydrogen bonds. So this is also correct. Option D is saying glycogen is a beta-1,4 glycosidic bond, which is absolutely wrong. In the glycogen, whenever we say the glycogen, there is a straight, suppose this is the one glucose, this is another glucose. Can you tell me what is the name of the bond? Yeah, the bond between two glucose molecule in the glycogen in a straight chain. Can you tell me, this is alpha bond, not beta bond, first of all, and this is alpha 1,4 bonding. Okay, and if I want to draw again like another glucose molecule, okay, so I am drawing another glucose molecule, 3,4 glucose molecule I have drawn here. Okay, so, and if I want to add it in this way, again I'm adding this. Okay, I'm adding this. These all are alpha bonds. Okay, alpha 1,4 bond. But if I want to branch it, like another new branch, you are getting another new branch you are forming here, another new branch. These branches are formed with the help of alpha 1,6 bond. These are formed with alpha 1,6 bond. So two type of bonds you can see. One is alpha 1,4 bond, another is alpha 1,6 bond. Alpha 1,4 for lengthening the chain, alpha 1,6 for the branching of glycogen. Okay, so this is called glycosidic bond, obviously, because they are formed in between glucose. But this is not beta. Beta is something wrong. This is alpha, because beta bond is present, I think in two cases you will find beta bond. Number one, in case of lactose. In case of lactose, I think lactose is nothing but the galactose plus glucose. Okay, galactose plus glucose. There you will find there you will find definitely, you know, this bond which is called beta bond. At the same time you will find beta bond, the popular example most of you knows, and that is cellulose. So cellulose and lactose, these are two compounds as per that you should know, where you can find beta bond. Okay, cellulose is a polymer which is homopolymer, homopolysaccharide. Lactose is a disaccharide. Clear, everyone? Okay, so the next question on the screen, question number 5 on the screen. Okay, question number 5 on the screen. Let me find how many of you can answer this question nicely. Question number 5, answer this question. Find out odd about the protein packaging. Find out the odd about the protein packaging. Okay, this is protein packaging is a very, very important concept. Okay, you have to definitely answer this question, protein packaging. So you tell me how many which all, you know, the out of these 4 statements, 3 statement is correct, 3 statements are correct with respect to protein packaging, but 1 statement is wrong. So you have to mark which one is wrong about protein packaging. Okay, you tell me, and people are answering already. So okay, this is a very important concept, very fundamental concept, and I'm pretty sure you can answer this question nicely. Okay, so Rupsha has answered it D, Shreya answered it D, Arnob D, Mihir Josh D, Ombika Dash D, Sudipta D. Only okay. Okay, others? Okay, Tina Ghosh. Okay, you are also D. Okay, what about others? Okay, all right. Ohana is saying also D. So let me explain these questions. Okay, Anindita, you are also saying D. Dipti is also saying D. All right. So the first statement, if I explain, chaperone is the protein, or chaperone, whatever you say, chaperone or chaperone is the protein which helps in protein folding. You know, like this is a protein, I guess, if I draw this protein, though I have drawn the tertiary structure of the protein, but if you want to package it now, you need another help. You need another help of another one, which is called chaperone protein. It's itself is also protein, and this protein will help you in packaging of the proteins. Okay, so this is the correct answer. Secondary structure, the protein has only the right-handedness. This is the NCERT statement, beta. This is the NCERT statement. This is not my statement, and this is a true statement that whenever I am saying the alpha helix version, you don't know alpha helix is the right-handedness. Okay, this is called right-handedness. You can see like it is just winding up clockwise, so right-handedness, and it's correct statement. Okay, so this is the true statement, this is the true statement. While you are solving this type of question, for your better understanding, they have asked you the odd one. So I have found many students, they know the question, but they don't have the technicality, and they don't have the proper technique to answer this question. If you are getting confused which one is true or false, you just write true and false while you are solving. Anyways, option C, enzyme is 3D structure, absolutely correct, and tertiary structure of the protein. This is a very, very important information, that enzyme, which is a functional protein, actually enzyme is considered as a functional protein. It is definitely 3D structure. It contains not only the hydrogen bond, it contains not only the hydrogen or covalent bond, but it also consists of many types of bonds like van der Waals forces and other forces. So tertiary structure of the protein is absolutely correct. And hemoglobin, enzyme, LDH are tertiary structure of the protein too. Okay, now I want to tell you, though all of the enzymes are tertiary in structure, but LDH, lactate dehydrogenase, is such an enzyme which is actually quaternary structure, and hemoglobin is also example of the quaternary structure, and these are most popular example of the quaternary structure as per your NCERT. So the option D is wrong. That's why option D will be the correct answer. Now let me explain that why hemoglobin, LDH, these are quaternary. It's very easy, you know. If you remember the structure of the hemoglobin consists of heme plus globin, and you know there are 4 globin chain, two alpha chain and 2 beta chain. That's why whenever you are finding multiple peptide chain in a structure, be sure it would be a quaternary structure. In the same time, if you see the LDH, in LDH, it also consists of different unit protein. Okay, unit protein, like it consists of heart unit, it consists of muscle unit. Okay, I just want to tell you, these are made up of few unit of peptides. This unit of peptides will ultimately form the LDH. So LDH is a very exceptional enzyme because First of all, it is also considered as isoenzyme. I think you know the concept of isoenzyme. Okay, isoenzyme is something, I mean, in your body, LDH, suppose in your body LDH is present, which are of 5 type, 1 2 3 4 and 5. Okay, suppose in your body, in liver, one type of LDH is present, in heart another type of LDH is present. So LDH can be present in 5 types, but what is the similarity? Similarity is that all LDHs are actually, the basic structure is actually same. Basic structure is same, I have told. Okay, so that's why they are called isoenzyme. So this is one point from lactate dehydrogenase MCQ. Second point is, it is actually quaternary structure because it consists of your heart unit and muscle unit protein. Okay, don't think in this way that heart unit is like those which are only present in heart. No, they are actually just name. We can also say it as a RAM unit and SHAM unit, but we have told it as a heart unit and muscle unit. Okay, don't be obsessed with the heart unit and the muscle unit. You just focus on it that LDH is quaternary protein because it consists of multiple chains of peptide, number one, and number two, it is an isoenzyme too. Okay, all right, next question. Okay, the next question on the screen, the next question, question number 6. Okay, absolutely correct. Arnav absolutely correct, Ankita correct, everyone is correct. Let us explain the options. Question number 6. Okay, let us explain the question number. It's a very tricky question. Okay, if you are, it's a 5-star question. It's a 5-star question. Let me explain. It's a 5-star question. It's a very tricky question and it's a 5-star question. Let me find how many of you can answer this question. It's a very tricky question. Which of the following is direct part of DNA? Which of the following is direct part of DNA? Okay, which of the following is direct part of DNA? You have to explain this. Which of the following is direct part of DNA? I think most of you will answer this question. Okay, it's a very tricky question. The catch word, let me explain how to pick up this catch question. Okay, let me explain how to pick up this catch question. Okay, so you tell me this first one. The catch question, first you have to know, in the question we have asked about the DNA. Okay. Second, you have to think about direct part. This is another catch. And then you have to choose the options actually. So tell me. Okay, let me explain now. Let me explain now because it's a very fundamental question. Okay, all right. Listen, when I'm saying you that it's a deoxythymidine, when I'm saying you it's a deoxythymidine, first you think about that whether it's a DNA or RNA. Yes, it is deoxy, so it's correct that it is DNA. Next question, I'm saying you thymidine. Okay, thymidine. Okay, you have to think about that. What is the nitrogen base known as? That is known as thymine. Okay, beta. Chalo. If it is thymine, beta, you know, when I'm adding sugar to it, which sugar, beta? You know, pentose sugar. Okay, pentose sugar. In DNA, definitely I can say this is a deoxyribose sugar. So I'm writing this DR sugar, deoxyribose sugar. So what you will get, beta? Thymine plus deoxyribose sugar. What you will get? Nucleoside. What is the name of the nucleoside? Thymidine. Thymidine. Now, beta, you tell me, it's your nucleoside. It's not nucleotide. I'm writing in short. It's only nucleoside. It's not nucleotide. So is it a direct part, beta? No, it's an indirect part because we have to also add phosphate to it. And while we'll add phosphate to it, it won't be your thymidine. It would be your thymidilic acid. Okay, so if it is your thymidilic acid, then it should be so the correct answer is not thymidine, it is thymidilic acid. So this option is actually wrong. Next question, uridylic acid. You know, uridylic acid means uracil. You know, it is not present in DNA. So straight away you can cross this option. If in option C, if I say cytidilic acid, cytidilic acid means, you know, this is cytosine I am saying. So cytosine I am saying, all right, but I am saying cytidilic acid. It is also correct because cytosine then nucleoside version is called cytidine, then its acid is called cytidyllic acid, which is nucleotide. But the problem with here, the deoxy part is missing, so it is again not a direct part. This is again wrong. So the correct answer is, now let me find, no one is correct. No one is correct in this question. The correct answer is this one. Here I am the winner only. This one. So now let me know how many of you, no one is correct. So everyone is bowled out right now. So you just let me know that whether you have understood the concept or not. If not, then I will explain again. Okay, okay, someone is saying, I think, okay, so people are saying, okay. Okay. I think it's fine now. Is it everyone? Is it clear, beta? Is it clear? Is it clear, beta? Yes or no? Let me know. Can I proceed forward? Okay. All right. This is a very fundamental questions, and I expected this question correct. Okay, I expected this question. Everyone will make this question correct, but okay, no issues. I think now you will not make mistake on this question. Okay, all right. So we can proceed forward. On the question number 7, this is another question which is based on a very basic concept, and it's again a question on DNA, and it's a previous year question. But here I just want to tell you how to choose the assertion reason question. Though very soon I will also upload another video on assertion reason, how to attempt the assertion reason. You know, being a NEET paper now, you will not be asked many assertion reason question. But yes, it can happen, as AIMS exam is also merged to the NEET exam. So few assertion reason question, like 4 or 5 assertion reason question might be asked. So you should know how to approach assertion reason questions. But before that, let us read this question together. If X is a DNA consists of 20% of adenine and Y is another DNA consists of 30% of adenine. Okay, all right. So we only know that this is a DNA which consists of 20% and Y is another which consists of 30% adenine. Now what we have to pick up actually? What we have to actually pick up? The assertion is DNA X has a higher melting point than DNA Y. The reason is Chargaff base equivalence says no one purine binds with one pyrimidine with hydrogen bonds. Okay, they should be bonds actually, because either 2 or 3. So the correct one is bonds. Okay, all right. So you just let me know. Okay, what is your answer? So the option A is assertion, reason, both are correct. B is assertion is correct, reason is wrong. C is assertion, reason, both are correct, but and reason also explain assertion. And D is assertion, reason, both are wrong. Both are wrong. Now explain. Now let me know what's the correct answer. Let me know what's the correct answer. All right. What is correct answer, beta? Okay, people are answering C actually. Okay, let me explain then. People are answering it as C. Okay, all right. Okay, so listen, in case of DNA number X, what is the percentage of adenine, beta? 20. If adenine is 20%, beta, what is your percentage of your AT? So it will be also 20%. Okay, so in case of X, DNA X, if A is equal to 20, definitely T will be 20. So our calculation is saying C is equal to G is equal to 20, 20, 40 is gone. So 60 remaining, 60 divided by 20. So 60 divided by 2, so it will be 30%. So C and G, they are present in 30% quantity. All right. In case of Y, this is DNA B and it contains 30% of the DNA, 30% of the adenine. So if it is 30% of the adenine is there in case of X, in case of Y, if your adenine is 30%, thymine is also 30%, and cytosine is equal to guanine is equal to 30, 30, 60 is gone. So 40 remaining, 40 divided by 2, so 20%. Okay. Now I want to say you something. A DNA stability depends on the number of triple bonds, and the triple bond presents in between only cytosine and the guanine. So if a DNA contains more triple bond, that DNA is more stable and the melting point for that DNA is high. So my first DNA contains 30% triple bond, like, you know, like CG, they have proper triple bond. So in the X, definitely they will have higher triple bond than Y. All right. So that's the reason that they have the high melting point. And how we can explain this? Chargaff base equivalence says one purine binds with one pyrimidine. Now, how to find that? Actually, most of the people, they are confused why whether this correctly. Okay, first, you know, both of the statements are actually correct. Chargaff base equivalence says one purine binds with one pyrimidine with hydrogen bond. This is correct. This is actually correct. So both statements are true. Okay, we don't have any doubt in it. But the question is, if both the statements are true, are they explaining each other? So then we have to read the question in this way. DNA X is higher melting point than DNA Y because Chargaff base equivalence says one purine binds with one pyrimidine with hydrogen bonds. This is not directly explaining your assertion. So though both statements are correct, but they are not explaining. The reason is not directly explaining the assertion. If I give you the reason like this, if I give you the reason like as per the Chargaff base equivalence, DNA Y containing more triple bonds than DNA X, then it would be a direct reasoning. Okay, and this direct reasoning is important if you want to tick the option C. So we have to take both the assertion reason are actually correct. This is actually the correct statement. Okay, both are correct. Yes, both are correct. This is the correct statement. Both are correct. C is not the correct answer. A is the correct answer. Let me find. Okay, Arnav, you were correct. Shubhadeep, you were wrong, beta. Anindita, you were correct. Shomramita, correct. Arnav first, or okay, Arnav is every time. Ananya, you are correct, beta. Onkita, you are also correct. Shayandeep, you are also correct. Srikanto, you said it's 6C. No, it's wrong. Actually, they are not mutually explaining each other. Okay, so assertion reason, whenever you are attempting assertion reason, the reason should be direct explanation of your assertion. All right, beta. Chalo. The next question, number 8. In the question number 8, the question is asking what is the correct relation between two successive bases of DNA. Okay, all right, nice question. It is again the question on DNA. So if I draw a DNA, you know, this is, if I draw a DNA in this way, now you say me, you know, whenever the DNA is starting its journey, okay, whenever the DNA is starting its journey from this point and it is coming up to this, it is actually crossing 360 degree. Okay, because it's a 3D structure. You can find in this way. If you can see me, just follow me what I'm trying to actually tell. Like I have if I have a DNA, like this is suppose I consider this is the staircase, and I mean this is the base of the DNA, nitrogen bases. Okay, stairs. So what do you have to imagine, like a you know, like if this is the point, and from where we are just winding it like this way, and it is coming in this. So here this is the 360 rotation, because this point, this started from this point. Okay, and again it is like moving 360 and it is coming again to the same position. Okay, and the same time it is also covering distance. Just at the same time it is also covering some distance to the, with respect to the axis. This is called pitch of the DNA. You know, that is called pitch of the DNA. Okay, so what I'm trying to say, that this is a complete turning of a DNA where they complete its 360 degree rotation. And here you have to remember definitely how many nitrogen bases you can expect in this position. All right, beta. So the nitrogen bases, what you can expect here is actually 10. Okay, in 360 degree you can expect 10 if I consider a BDNA, which is actually a typical DNA, is called BDNA. So if I say the every DNA, what is the angle between every DNA? This what is the angle between every base? If I say this base and this base, 10 base pair, they cover 10 base pair, they cover 360 degrees. So one base pair covers 36 degree. So absolutely correct, this statement. Next statement travels a linear distance 3.4 nanometer, travels a linear diameter of 3.4 nanometer. Okay, this is something you have to remember actually, which is very, very important. The 360 degree rotation, the pitch, 360 degree rotation, the 360 degree rotation, what is the pitch of 360 rotation? You know, this is something you have to remember. This is 34 angstrom. Okay, 34 angstrom. So I am saying 10 bp is equal to 360 degree rotation is equal to 34 angstrom. So 1 bp is equal to 36 degree rotation is equal to 34 divided by 10, I mean 3.4 angstrom. And I think you know that angstrom has to be divided by 10 if you want to get it in nanometer. So it will be your 0.0 34 nanometer, 0.34 nanometer. Okay, all right, 0.34 nanometer. Okay, all right. The next one, which is a very, very important. Okay, people are answering 1218. Okay, let me find. Okay, I will also say you what is the correct answer. Okay, don't be misguided with this @ sign. Okay, don't be misguided with this @ sign. That is something I want to tell you. Okay, well, let us solve this question first. The correct relation between two successive bases of BDNA. The first one is correct, that I have already explained. The second one is 3.4 nanometer, travels a linear distance 3.4 nanometer, two successive bases. No, this is wrong because it is 36. If I say that 34 angstrom, 3.4 angstrom for 2, but not 3.4 nanometer. Okay, that's why it becomes wrong. Turns in right-handed direction. Turns in right-handed direction. So yeah, BDNA is right-handed DNA, ZDNA is left-handed, so it is also correct. Okay, I think the question can also come wrong. Bases are attached independently with sugar phosphate backbone with carbon bond. Bases are attached independently with sugar phosphate backbone. Okay, all right. So you know, when I am saying this, so let me explain this one. If I am say this is the DNA, you know, if this is the DNA, if I say this is the DNA, so the nitrogen bases are in this way, they are mutually attached together with hydrogen bond. But when I'm saying how they are attached with the backbone, then obviously it is carbon bond through which they are attached with the backbone. So it is carbon bond with which they are attached to the backbone. So this one, I think, you know, let me again mark. So this is, you know, this is the sugar phosphate backbone, the yellow that I have marked. This is the sugar phosphate backbone. Okay, all right. And this is something, let me mark. Yeah, this is the red color I am marking right now. This is the nitrogen bases. Okay, and they are nitrogen bases. Like say this is the adenine, this is the thymine. They are mutually bonded together with double bond. And I want to also say you that how they are attached with the sugar phosphate backbone. This is the carbon bond. Okay, sometimes this is something, it might also happen like nitrogen carbon bond, but not all time carbon bond. Sometimes they may also have carbon nitrogen bond as well. So now the question is saying that the carbon bond. Okay, so what I want to say you here, which one is correct? This is not carbon bond. Actually, this is carbon nitrogen bond. Okay, this is the carbon nitrogen bond. I mean the nitrogen of the nitrogen bases. I think you know the nitrogen bases structure. I think you know the nitrogen bases structure. This is the structure of your like 1 and 3 position if the nitrogen is present. This is something called, you know, this is something called your, which one? We can draw two structures. One is purine and that is pyrimidine. If you say about the purine, it is the heaviest, pyrimidine is smallest. So this is the structure of the pyrimidine, where the nitrogen is present in first member of the ring and the third member of the ring. Okay, and this nitrogen, they will form bond with the sugar phosphate backbone. I mean with this. Okay, so this will be an NC bond. This is not a CC bond, rather it will be NC bond. Okay, that's why it is wrong. And turns right-handed directions, yes, this is actually also correct. So we have to mark actually both option A and option C, both. Option A and option C, both. Let me find people are answering it. So those who are answering option A and option C, both are correct. Okay, so you can mark option A as well and option C as well. Both are correct, beta. Both are correct. Both are correct. Okay, option C, option A. Okay. So what you were saying, option C, option A. Okay, all right. Yeah, both are correct, beta. Okay, chalo. Anuna Chaudhary is saying A. Yeah, A is obviously correct as well. Now tell me question number 9. Question number 9. It's a question based on the concept of your fat. If you know the concept of fat, this would help you. Okay, the question is oil has low melting point. Okay, that is the first option. Option B is saying cell membrane has phospholipid, surfactant is made up of DPPC, and option D is saying stearic acid, palmitic acid, this has one double bond. Okay, so let me know. Okay, people are answering this question. So which one is false? Oil has low melting point. Okay, now right answer here. Okay, so this is the question against question number 9. So I am asking to type the answer against question number 9. Question number 9, I have already typed question number 9 in the chat box. If you want to answer, those who have answer, people are answering D. Anuna Chaudhary answering D. Okay, listen. Okay, so you keep answering and I will tell you who one is correct and who one is wrong. Okay, so you know the oil. Okay, oil has a low melting point. This is correct actually, because this is the difference between oil and wax. Wax has high melting point, that's why in atmosphere it is solid, but oil is low melting point, that's why even in the atmospheric temperature they are liquid. All right, the second question, cell membrane has phospholipid. I think this is a very easy statement, you already know they are phospholipid. This is the glycerol, this is now fatty acid tail. And if you want to, and I think you also know the structure from since your childhood, that the glycerol, okay, this is the glycerol structure, and you know with this glycerol structure, this is glycerol, I think you know already, this is CH, H, H and H and H. Now the question is you have to add a fatty acid, you know fatty acid is COH, okay, with like R, then you will find ester bond, 3 ester bond, that's why it is called triglycerides. And you know this is called fatty acid tail, so these are called fatty acid tail. And one thing I want to tell you here 2 fatty acid is enough, tail enough, but with 1 definitely you should have the phosphoric acid or phosphate group, so that's why it is phospholipid in the cell membrane. Absolutely correct, absolutely correct. Okay, surfactant is made up of DPDC. I think most of you know what is surfactant. Surfactant is something which is present in our lung alveoli, and this surfactant is released by a particular cell which is called type 2 cell of alveoli. It is also synthesized by the Clara cell of other airway, like your something is called alveolar duct, something is called respiratory bronchiole. There is Clara cell present, and in the alveoli you have pneumocyte 2 cell. So in the alveoli you have pneumocyte 2 cell, in the respiratory bronchiole and in the alveolar duct you have something which is called Clara cell. Okay, and these cells are actually secreting your surfactant, which is your MCQ. This surfactant actually it's nothing but a layer of fat. Surfactant is a layer of DPDC. What is DPDC? Dipalmitoyl. That is, I mean, it is made up of a fatty acid which is called palmitic acid, phosphatidylcholine. Okay, and it actually prevents the inward force of alveoli, which is called collapsing force, which is called also surface tension. So it will decrease the surface tension. So surfactant will help. Why it is named as surfactant? because it will decrease the surface tension. So it is also correct statement, and it is made up of DPDC. It is correct statement. Stearic acid and palmitic acid has one double bond. Absolutely wrong. Stearic acid, palmitic acid, they are saturated fatty acid, so they don't have even a single double bond. They have only single bond. So that's why option D is the correct answer. I think most of you have answered it correctly. Okay, Arnav Hajra answered correctly, Ambika answered correctly, Shubhadeep answered correctly, Srikanto answered correctly. And then again, okay, everyone has answered. Show me, fantastic. Show me, you have also answered correctly. Shayandeep, Dipti Pal, Anandita Chatterjee, Onkita Ghoshal, Onkita De, Ohana. Okay, fantastic answering, fantastic. It is an easy question, but I'm happy that you have also made it correct. Okay, next question. Next question is a very, 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 very important question, and it's a 5-star question. It's a 5-star question. Okay, this question is based on a concept which is called secondary metabolite, secondary metabolite. This question is based on a concept which is very important, which is actually called secondary metabolite. Okay, secondary metabolite. I think all of you have read the there is a chart in NCERT. What are the secondary metabolite? I will not write everything. I just want to tell you there are a few pigments which are secondary metabolites. There are a few alkaloids which are secondary metabolites. Okay, so there are a few. Lectin is also secondary metabolites. So you have to answer which one is not secondary metabolite. Okay, morphine, it's alkaloid which is secondary metabolite. Okay, this is lemongrass oil is an essential oil. Okay, it is secondary metabolite. Oil is also considered as secondary metabolite, but never ever you have heard that cellulose is a secondary metabolite. That's why the correct answer, okay, just let me, okay, sorry for the wrong answer. If I say it, then definitely it would be your, listen, let me explain again, please. So this is oil is something, yeah, oil is something which is always primary metabolite. Okay, oil, if I say only oil, it is primary metabolite. If I say grass oil, then secondary metabolite. But cellulose is something, and cellulose is also something which is considered sometime, though I know cellulose is made up of glucose and glucose with beta bonds, beta 1,4 that you already know, but it is also considered as secondary metabolite. Okay, it is also considered as secondary metabolite. If you have any confusion, you can also check. Yeah, cellulose is a polymeric substance. Obviously it's a polymeric substance, and that's why we actually consider it as a secondary. Listen, the concept of secondary metabolite is actually which is not directly produced by body. Okay, like if I say, I mean, not directly available, not available in a straightforward way. Like if I say the biomolecule which are actually monosaccharide, if I say amino acid, if I say these are something, or if I say lipid, these are directly available. Okay, but secondary metabolite means you have to synthesize again from the primary metabolite. Okay, cellulose is something which we have to synthesize from the glucose. That's why it is secondary metabolite. Correct answer is oil. Question number 11. Which of the following is not strictly macromolecule? Again, and repeatedly I answer this question. Okay, and you have to also know the concept of this question. Which of the following is not strictly macromolecule? Which of the following is not strictly macromolecule? Yeah, oh, no, you are correct. Polymeric substance, rubber, gum, cellulose, these all are secondary metabolite. Cellulose is a secondary metabolite. Rubber, gum is a secondary metabolite. Which of the following is not strictly macromolecule? The central concept behind this question is whenever we are saying macromolecule, they should satisfy either of the criteria actually. One criteria is their molecular weight actually should be more than 800 Dalton. And second criteria is, if I want to say any one macromolecule, definitely they should react with acid. Okay, they should react with acid. Okay, they should react with acid, and when they are reacting with the acid, basically they should form some insoluble portion, insoluble fraction. Okay, if they are producing insoluble fraction, or they should have 800 Dalton. So all of this, protein, nucleic acid, polysaccharide, okay, they are macromolecule actually because they satisfy both of these criterias. But if I say about the lipid na beta, if I say about the lipid, in case of lipid, the molecular weight most of the time is less than 800 Dalton. So this condition is not satisfying. But the second condition, if you react your lipid with acetic acid or anything else, any organic acid, okay, they will give you an acid insoluble fraction. Acid insoluble fraction. All right. So that's why the correct answer is option D, that we cannot consider lipid as a strictly macromolecule because their molecular weight is less than 800 Dalton. Question number 12, which is a very, very important question again. Find the wrong match. Wrong match. If you are making this silly mistake that you forget to mark a word wrong when you are answering this question, then I would request please mark under this wrong option. So under the word wrong. So it is a wrong match, you have to mark it. If I say trypsin, it is a pancreatic enzyme that you already know, which is a proteolytic enzyme. So absolutely correct. If I say about the, okay, let me find how many people are answering this question. It's an easy question, beta. Why you are so, okay, let me type this in the chat box. Yeah, so question number 12. Okay, so there is question number 12. Okay, how many of you are answering question number 12? Okay, question number 12. So I want to answer against question number 12. Okay, I have already started receiving the answer. Okay, question number 12. Okay, insulin is a chemical substance of chemical coordination. I think you will also agree because it's a hormone, so obviously it will be a substance of chemical coordination. If I say about the GLUT4, then you have to know something, the concept, what is GLUT4. So then you have to know what is a GLUT. GLUT is a glucose transporter. If I say the glucose transporter it's a bit different from the sodium dependent glucose transporter because GLUT they do not require any type of support from anyone. But what is the difference of GLUT4? GLUT4 is something which will prevent you from diabetes. You know, like my blood is having a huge amount of the glucose, but it should enter in the cell. Which cell? Preferably it should enter in the muscle cell or it should enter in the lipid cell. These are two cells, adipocyte cell, one is lipid cell, or adipocyte cell, one is muscle cell, where my glucose should enter once I am making a pool of glucose in my blood. Okay, and this is somewhere done by a particular glucose uptake glucose channel, which is actually called GLUT4. So this is your GLUT4, which will help for the entry of glucose inside the cell. All right, but I just want to tell you there are many GLUT. If I say GLUT1, GLUT3, GLUT7, GLUT6, that can be present in many cells, they are present. But when I'm saying the GLUT4, it is present only on the muscle and not liver. This is the lipid cell. I can also lipid cell, or you can also say like adipocyte cell, fat cell. You can say fat cell. So this should be present in the fat cell. Okay, this should be present in the fat cell. Okay, just let me answer it. Okay, fat cell. So that's why this is obviously wrong statement. And I want to add one extra information to this question actually, which is very phenomenal information for MCQ, in the world of MCQ. This activity of the GLUT4 is dependent by a hormone. Can you tell me the name of the hormone, beta, please? The name of the hormone is actually, you know, it is called insulin. So when you are taking insulin, then we have adequate insulin, so we don't have any problem. But if you lack insulin, then you have to then what would happen, that you have to take artificial insulin from outside, which will be produced by, you know, recombinant DNA technology, which was first produced by Eli Lilly. This is something information that we have already discussed in the biotechnology chapter. Here I want to tell you, basically this insulin will act on GLUT4. Okay, and when it will act on the GLUT4, your sodium glucose will enter in the cell, and it is present in the fat and muscle cell only, so not in all cell. This is the wrong statement, not all. And option 4, antibody, it is a protein or peptide, you can say, fight against infectious agent. Yeah, this is something basic statement you already know. So correct answer will be C. Now let me find how many of you are answering correctly. So Deepika Dash answered is correctly. Oh, nice, Deepika. Arnab answered correctly. Ankita answered correctly. Anindita Chatterjee answered correctly. Ohana answered correctly. Shreya answered correctly. Shoromita answered correctly. Ankita Ghoshal answered correctly. Shubhadeep Bose answered correctly. So the next question, question number 15. I'm writing in chat. Question number 13. So next question, question number 13. I have already written in the chat question number 13. Now I will be waiting for you answer and the question on the screen. Question: wrong about the carbohydrate. Okay, wrong about the carbohydrate. Which of the following is wrong about the carbohydrate? So if I say the wrong about the carbohydrate, then inulin is a polymer of fructose. That is the first statement. Second statement: starch can hold iodine. That's a nice and interesting statement. Cellulose has helical structure, can hold iodine. That's another nice statement. And chitin is a homopolymer found in the exoskeleton of the arthropods as well as the fungal cell wall. This is again nice statement. So there are 4 statements which are given with respect to the carbohydrate, and people are already answering as option C. Now let me explain, because okay, but other keep answering this question. Inulin, if I say about the inulin, it's a polysaccharide and which is polymer of the fructose. Absolutely correct answer. Which is substance which is used in the kidney function test or renal function test. Well, if you want to test your kidney, you have to, and if you want to estimate your GFR, glomerular filtration rate, you should take help from this substance which is called inulin, and that's why it's a popular question, and it is available in the root of some plants. I think you know the name of those plants from where you can avail your inulin. That's something is called shotomuli, there's something called asparagus or dahlia. Okay, now starch can hold iodine. This is absolutely nice statement, because if I draw the structure of the starch, let me draw the structure of the starch. The starch, you know, it's a branching structure. Okay, you know, I have already told you glycogen and starch, they have branching, though glycogen has more branching because it is present in the animal body, so it should actually branch more to configure it inside the human or animal body. But as the plant size is more, you can remember in this way that starch is actually less branching, glycogen is very branching. But whatever be it your starch or glycogen, make sure you know that they are branching. So as they are branching, so definitely they can hold iodine in their branch, and that will give the actually the characteristics violet color. Though the color of the iodine itself is brown, you know, but when they will be present inside, you know, the trap of glycogen, they will give you the violet color. So this is something which is an absolutely right statement. Okay, absolutely right statement, that cellulose has its helical starch can hold iodine. Absolutely correct statement. Cellulose has also helical structure and can hold iodine. No, this is wrong, because if you have seen the structure of the cellulose, it's the straight chain compound, it will not form the branching. So how it will hold the iodine? So this is a wrong statement. Okay, and chitin is a homopolymer. Yeah, this is a NCERT update. I want to tell you everyone, and to tell everyone, this is homopolymer. This is a correct statement. Okay, beta. In previous edition of the NCERT, there was a mistake that it was mentioned as a heteropolymer, but this time they have updated it as your homopolymer, and that's a correct statement. So chitin is homopolymer found in the exoskeleton of arthropods. Absolutely correct. Also it is found in the fungal cell wall, and you also know chitin is present not only the exoskeleton of the arthropods. Okay, this statement is definitely true, but I just want to add on extra points. You know, even in the reproductive organ of the cockroach, even in the elementary canal of the cockroach, you have chitin as well. That is something I want to tell you. So so absolute wrong statement is option C. Absolutely wrong statement is option C. So the next, and I guess this is the last question, is very interesting questions of the day. That is something. Okay, before that, let me announce okay, who were correct. Shubhadeep Ghosh was correct. Shomi Mondol correct. Deepika Dash, Shreya Paja, Tina Ghosh, Anonnam, Ankita De, Ankita Chatterjee, Ohana Shaha, Ankita Ghoshal, Diptipal. Okay, these people were correct. Okay, now the last question of the day. Identify the number of enzymes with heme as a prosthetic group. Okay, please do remember, whenever you know in an enzyme, whenever you are saying the enzyme beta, please do remember there are two parts. One is protein part, one is non-protein part. Okay, So if I say about the protein part beta, if I say about the protein part, this thing you have to remember when I'm saying the protein part, this is called apoenzyme. Okay, and when a non-protein part is actually known as cofactor. Okay, now this cofactor is again two type. If you find your cofactor is tightly attached or loosely attached, if you find it is tightly attached, then you can see it as a prosthetic group. And is loosely attached, you can say it as coenzyme. Okay, you can say coenzyme. Now and whenever you know beta that when you will find both the enzyme, when you will find both the protein and the non-protein together, what is its name beta? This is called hollow enzyme. This is something called hollow enzyme. Okay, I think you know this is called hollow enzyme. Clear everyone? Clear everyone? Okay, now let me tell you there are names like cytochrome, hemoglobin. Okay, let me highlight these names: cytochrome, hemoglobin, myoglobin, catalase, and peroxidase. We can clearly mention here that there are 5 chemical compounds we have mentioned. We have to know, this is an NCERT based question. Okay, always remember, heme is a porphyrin compound which is synthesized from the succinyl CoA of Krebs cycle with addition of glycine. And finally, I want to tell you, after the heme synthesis, once the heme is synthesized, they can be a part of all these compounds which are being mentioned. Cytochrome, that is a part of electron transport chain. Hemoglobin, you know, myoglobin is a muscle respiratory pigment, and you know, catalase is an enzyme, peroxidase is an enzyme. So there are, you know, catalase is enzyme, peroxidase is an enzyme, but cytochrome, hemoglobin, and myoglobin, these are not actually enzyme, they're proteins actually. But I want to tell you, for all these, heme is the prosthetic group. So the correct answer will be 5. Okay, only Shubhadeep is correct here. Only Shubhadeep correct, I guess. Yeah. That was the last question. Though I don't know whether I have written question 14 or not. So 14, Ananya Chaudhary is saying B. No beta, no beta, you were wrong beta. This is something you have to know. I know you people will make mistake on this question, that's why I told. No, C is also wrong, not 4. Why you are saying 4? This is 5. All these 5, myoglobin, catalase, you know, like cytochrome, peroxidase, all these contains your heme, which is actually present as prosthetic group. I mean, they are attached tightly. Again, I'm mentioning heme is, whenever heme is present, they are present tightly with its protein part. Okay, so heme is prosthetic group, always do remember, and they are present in your these compounds: cytochrome, hemoglobin, myoglobin, and catalase. Kazi Abdul Rizwan, yes beta, this should be D. This should be D. All right, this should be D. 5 is the correct answer. 5 is the correct answer. So thank you for attending this session, and I guess we have solved 14 to 15 very nice question with its discussion, which will be helpful for you definitely. And for the next session, I will update in the Telegram channel, as well as executive will broadcast the messages as well. So stay tuned, prepare well, and all these questions, please note it down nicely. And the next live update you will get also in this YouTube channel. So if you haven't subscribed, you should subscribe actually. And thank you everyone for joining the sessions.